Now we are going to talk about the raw materials which are important for the process of photosynthesis, using which the plants are ultimately giving us the organic compound of glucose. Now what are the raw materials? Primarily there are four raw materials that you have to remember. First one is of course carbon dioxide that would get reduced to form the glucose molecule. Second one is water. Third one is sunlight, and fourth one is the chloroplast. This chloroplast is a cellular organelle, that is plastids which are exclusively found in the plants. You find chloroplasts. So these are the four raw materials which are going to be required when the photosynthesis has to take place, and this lesson is going to focus on these two so that by the end of the lesson you know what is the role of these two raw materials. These are basically the reactants in the chemical reaction which ultimately give rise to glucose. Talking about the first one, that is carbon dioxide, it is very clear that the plants, they take up the carbon dioxide from the atmosphere through the pores which are present on their leaves known as stomata. There is gaseous exchange that takes place. The atmospheric oxygen is always inside the leaf. There is always the scarcity, or you can say there is thirst for carbon dioxide, and it gets diffused inside and it is taken up along with the air that goes inside the leaves. This carbon dioxide would get reduced. Okay, so basically what is photosynthesis? It is an oxidation reduction reaction where the carbon dioxide will get reduced. Now this part of understanding the process of photosynthesis involves your knowledge from chemistry, where you study redox reactions, talking about the reactions at the level of hydrogen ion exchange. Alright, so the hydrogen ions would be taken up by the carbon dioxide. That means it would get reduced. There is reduction which is taking place, and when something gets reduced, that means some reactant has given its hydrogen ions. So that particular compound, or you can say reactant, gets oxidized. So there is simultaneous oxidation and reduction which is taking place, which gives rise to glucose. Now coming to the second raw material, that is water. You have to keep a few things in mind, that when we talked about the reaction in the introductory lesson, we had discussed that it is basically the water which is giving the protons, alright, which is responsible for reducing this particular carbon dioxide inside the plants. Now talking about the reduction and talking about the raw material, it was not clear in the beginning that it is the water that gives the hydrogen ions and in return evolves oxygen. It was considered in the beginning, like myself, when I was in the stage where you are right now, I used to think in the beginning that this oxygen gets evolved, this particular oxygen gets evolved as it is, and this water comes and joins with carbon. It is not the case, and it has been it has been proved by a few scientists. Now we are going to see chronologically how it happened. There was a scientist named Van Niel, the first pioneered person who thought about a particular reaction, and in the end he came to a conclusion that water is the proton donor. Alright, how he came to that, we are going to discuss that as well right in this lesson only. In 1931 he discovered this. After like a gap of 6 to 7 years, Robin Hill was another scientist who saw that there is a lot of weight in what Van Niel has said, and he proved it experimentally using a few compounds. Again, that would be a chemical reaction. And last one would be Rubin and Kamen. They were also involved in this particular understanding. Okay. Now they came in 1934, but there was their principle was of the principle was based on radioactivity. Okay, we're going to see how. talking about Van Niel, what he did is that we have to establish from understanding this that it is the water which is the source of proton, and it is the water which gives rise to oxygen. So either we can concentrate on proton donation, or we can concentrate on liberation of oxygen, because liberation of oxygen is an easier concept to track experimentally. So we particularly concentrate on this aspect. Alright, now what Van Niel had observed, that there was certain bacteria which were dependent, they were not requiring water. Okay, there were certain photosynthetic bacteria, they were not requiring water. So when they are not requiring water, they are not going to evolve oxygen as well. So that is what he observed, that those photosynthetic bacteria, they were dependent upon some sulfur source. Alright, please pay attention that there were some photosynthetic bacteria. That means they are performing photosynthesis. That means the carbon dioxide would be fixed and glucose would be formed. That means this thing would be formed, this particular thing would be formed, but they do not require water. Alright, instead they depend upon another thing, that is this sort of compound. Okay, now by accumulation of sulfur as a byproduct that was observed in his experiment, he established that sulfur comes as a byproduct in his experiment. The bacteria is using this particular proton, this particular compound as source of proton, and sulfur is getting accumulated in the same way, because oxygen is not getting accumulated. Why oxygen is not getting accumulated? Because there is no intake of water in the reaction. We are getting the glucose molecule, we are getting the carbon dioxide getting reduced, but water is not involved. If the carbon dioxide is getting reduced, water is not involved, then how it is getting reduced? It is getting reduced by H2S. Now this H2S is giving its protons, and at the same time you are having sulfur accumulated. That means this particular compound gets oxidized itself. Alright, there is a certain compound which gets oxidized itself and reduces carbon dioxide. Okay, oxidation of H2S gives rise to reduction of this particular compound, right? Now this oxidation of water, that means the loss of protons from the water, gives rise to oxygen in those particular plants where water is used instead of H2S. I hope I have tried my best to make you clear, but in case still you're having a problem, I'm going to repeat it, that this person had to establish that it is not the carbon dioxide which gives oxygen, it is the water which is the source of oxygen that gets liberated, and primarily we are not concerned about what is oxygen source and what oxygen has to do. We are more concerned about production of this particular compound. Now this particular compound cannot be produced if carbon dioxide is not there. Those experiments are of lower class level. Now we have to deal with the reduction part. Alright, if the reduction is taking place, that means the carbon dioxide is picking up the protons, where does that proton supply come from? That comes from water. And that was established when instead of water, certain bacteria, they used this particular compound and they used these protons for reduction, and byproduct came as sulfur. So it was somehow established that ultimately it is water which is being used for providing the reducing protons. Alright, now then came Robin Hill, who proposed that in presence of certain Hill reactants, you have to remember the term Hill reactants or Hill oxidants. Hill reactants, Hill oxidants we can call. The Hill reactants were what? They were certain compounds which could be reduced. Okay. Now when the compound can be reduced, he used DCPIP, dichlorophenol idophenol. That was the compound that was used in his reaction, it is blue in color. Now see, this is to be established that something is taking up the compound, that is taking up the proton. Okay, now what would be taken care of? In the previous case, in the case of Van Niel, what was done, that water was missing. Carbon dioxide was there, of course illumination would be there, chloroplast was there, everything was involved. In this particular case, we are going to get rid of carbon dioxide. We are going to miss that particular aspect. We are not going to use carbon dioxide. In the absence of carbon dioxide, in the presence of water, in the presence of chloroplast, when a particular leaf is illuminated, and in the same culture, the culture that was used was chlorella. Remember this as well. Chlorella is an algae. So this chlorella was used, its culture was used. One thing was missing, that is carbon dioxide was missing. Now you have to establish that the protons are being provided by this particular compound, alright, and you have to establish that this particular compound provides the protons in the presence of sunlight to reduce something. For reduction, we have provided in the culture DCPIP, which is blue in color. Now what is done in the reaction is that this particular reaction, this particular scientist, he proposed that something is getting reduced, and that reduction is taking place by the presence of water. For that, he made the carbon dioxide absent in the reaction. What he took was, he took a leaf and he just get rid of the veins which were present. He just crushed that leaf, and instead he used chlorella. Don't get confused. That leaf particular is a demonstration that we often do. You just have to consider that he took chlorella. He provided with a compound that could be easily reduced, that is otherwise known as Hill's reactant. He provided this particular culture with a compound. This compound was blue in color. Now, he is aiming at reducing this compound. This compound has to take up the protons from the water. Now, that is what happened. When illuminated by the sunlight, the chloroplast and the chlorella, they took up the sunlight, they just took the energy, they passed it on to water. The water just took up that energy, it broke down, the protons were taken up by DCPIP, okay, the Hill reactant, and the blue color of the test tube, it goes away. Alright, the particular culture, it becomes colorless. That means that this particular reactant was reduced, okay, it was not in its present form as in the beginning. So this was Hill's reaction. Again, I am going to repeat it, that what was taken care of, that it was to be proposed in the end, and it was to be confirmed that water gives the protons. Okay, when water gives the protons, whatever is present, it is going to take up and get reduced. The protons would be taken up by something which would require those proteins. That means a certain compound which could be reduced. That means oxidizable compound, and that particular compound was this DCPIP. Now, carbon dioxide went missing in the reaction. This carbon dioxide being absent, what was taken care of, that this particular compound takes up the protons. Now, it is easy to establish at this stage that protons are given by water. Okay, they are given in the form of protons. It is not just the water that comes and joins with carbon dioxide. Oxygen is still getting evolved. Alright, in the end of the reaction, what we have come to as conclusion, that oxygen is still getting evolved. That means in liberation of oxygen, there is no role of carbon dioxide. In the liberation of oxygen, the primarily water is playing the role. That means this oxygen molecule that we talk about and it gets liberated, it comes from water. When we say that it comes from water, it is quite certain that the remaining part of the molecule would act as protons, and they would go and reduce this, as was seen in the experiment. So this was again proposed by Robin Hill, and in fact confirmed that oxygen is released from water, and water acts as the source of protons for reduction of carbon dioxide. Lastly, it was these two who had given the principle that the reduction of carbon dioxide takes place by emission of oxygen from water molecule that gets broken down. They used O18. Now O18 is radioactive oxygen, and they used the water which had this particular radioactive O18. So basically they used this particular, not like this, it is to be written like this. Okay, I wrote it differently. This was the radioactive water which was being used. Now it was observed after the reaction, again the same illumination, the chlorella algae was used, the chloroplasts of the algae, they were using water which was radioactive, and what they are going to give rise to, that these particular protons would be used up normally in the formation of glucose, and the oxygen that you are going to have, the molecular oxygen that you are going to have would be radioactive. So it is enough to prove that basically the oxygen is getting evolved from the water molecule, not from the carbon dioxide. So what happens to this water, that also you have to remember. And what is the role of water? If there is one molecule of water that is involved, you are going to have 2 protons and 1 nascent oxygen. So by evolution of one oxygen molecule, we have 2 water molecules. For evolution of one oxygen molecule, we will have 4 protons and 1 oxygen. That would be molecular 1. Now when 4 protons are released, because they are at this condition, we have 4 electrons which are also being released. So this is the raw material water that we have talked about, and its role, which was proven by a few scientists who had experimental demonstrations to show that it is ultimately water which acts as the source of oxygen as well as the source of protons, which come by splitting of the water molecule. The hydrogen ions are generated, molecular oxygen is being liberated. So this is the equation I am going to write over here, that the water molecules would break down to protons, they would break down to electrons of course, and the molecular oxygen would be liberated. And that's what we have dedicated this entire lesson to, that ultimately the liberation of oxygen is taking up from water molecule. Okay, and these two are the important raw materials for the process of photosynthesis. So this is all about these 2 raw materials. The next lesson we have dedicated for light and chloroplast. For that you need to watch the next